today I will be presenting a presentation about CRISPR as a new approach to cancer treatment. At first, I want to briefly talk about the history of the CRISPR and about who discovered this new technique. Two women scientists back in 2020, they worked together to discover this technique. Jennifer Doudna, which she's a biochemist, and Emmanuel Charpentier, which she's a microbiologist. They both discovered about the CRISPR-Cas9. It's a new method that removes and adds pieces of DNA, can alter any organism's type of organism's DNA, and also it's an exquisite precision. So what does the CRISPR stand for? CRISPR is clustered regularly interspace short palindromic repeats, abbreviated as CRISPR. It's a highly specific and rapid modification of DNA. What does this approach do is that it can make the change of the DNA from the genome. It can cut in part and add another one. So what is the importance for this? As I said, it can rewrite the genetic codes. It can rearrange the nucleotides again and add new pieces for it. So, for example, let's say we have a gene which is responsible for transferring the genetic disease of diabetes or cancer or let's say hypertension, because sometimes it's also a genetic disease. We can just remove that part which is responsible for it and add a new one which is healthy and cannot transfer it, and the family can be healthy forever when they marry and they make a child. CRISPR has so many advantages and benefits, but I will focus on three. The higher gene editing efficiency, it's very benefit that can change the sequences to a healthy one, as I talked about it. It can lower off-target effect. So what is a off-target is, for example, we're targeting to do the gene editing in a liver cell, which let's say the patient has the liver cancer and we want to change it. So we are targeting to do the change in the liver cell, but in previous methods it could go wrong and it could alter the muscle cell or the stomach cell, which was a dangerous procedure for that time. And also it has benefit that no DNA integration happens, because we are unlooping the DNA to the RNA to make the change for it. It prevents the unification for the DNA. I will explain this figure. It's called immunotherapy. It's a new type of therapy for cancer patients. It's targeting to help our immune system to fight off the cancer. Also to minimize the use of chemotherapy, which we all know it has so many side effects, but also this is a very expensive treatment. In America, it costs like for $132,000. It's very expensive, but it's a very efficient method. What is done is that we collect the blood from the patient. We will collect the T cells and isolate them, adding some receptors and particles of the cancer cell, which helps the T killer cell, which we all know the function that is to kill the cancerous cells. We will add it, and the T cell can easier recognize the cancerous cell and alter and kill it. After that, we develop the cells in laboratory. We multiply them, and then we give it back to the patient, and with this, we help the patient to cure faster. The role in treatment is that to edit the gene. How? I will just explain it by drawing a simple figure. Let's say that this is the DNA we have it. We have a sick part here. So let's say this part is responsible for offspring of the thalassemia. By the use for this method, by using the enzyme of Cas, we will just delete this or cut this off and replace a a new one to the DNA, and forever they will be get rid of this disease. Also, as CRISPR is expected to resolve the major challenges to improvement, that it can be a method to eradicate the genetic diseases forever, which is very efficient and benefit for the humanity. And I really want to focus on this, that how this approach is being done for cancer. So we have two kind of tools, which is the guide RNA and the Cas enzyme. We have the enzyme. It looks something like this. We call this the Cas enzyme. And it has something which is called the guide RNA. When I explain it here, you will understand better the figure. So how the CRISPR even was found? It was found from the nature. Like they noticed that when a virus is invading the bacteria, that we know it's called the bacteriophage, and the bacteria is just getting a part of the DNA and store it inside. When for the second time the virus is coming to the bacteria will know it that before I have the genome for the virus, and what the bacteria does is it will just cut down the genetic material of the virus, and it will rescue from itself. And then scientists were inspired by this method, and they tried to do it also for human. But as I said, it has two tools: the guide RNA here and the enzyme, the Cas enzyme. The scientists designed to the guide RNA is a mirror for the DNA, for the DNA of the gene to be the target. The target is like for the liver cell. Let's say that for the liver cancer, we plan to do the gene editing in the liver cell, which is when the enzyme and the target genes DNA are combined together, the Cas will just cut off the part and will add another one, another healthy one. But there is something that we maybe, as I explain it, it will go to another target cell, which is not the target, but the enzyme will go to a wrong place, which is not what we plan it for. Like it will alter the muscle cell, and it will cause another thing. Or at the same time, it may cause the healthy cells to be a cancerous cell. This is a very big deal. But the scientists are planning to tweak the the guide RNA able to be improved by intended targets. Like they're working on this to help the guide RNA specifically go to the target place, not to go anywhere else. Also, we can here use something to transfer the tools of the CRISPR safely to the cancer cells. How? By a virus. So we are modifying the virus not to harm us, but to transfer the tools to our cancerous cells. The tools are guide RNA, Cas enzyme. But how our immune system will deal with this? Because we know when the virus is a non-part of the immune system, it's in common way that, of course, the immune system will attack and try to get rid of it. My opinions are by immunosuppression, giving medications to the patient to suppress the immune system and not let the white blood cells to alter or kill the virus, which holds the tools of the CRISPR, or by giving chemotherapy before this therapy. So it may be a combined therapy, like for chemotherapy and the CRISPR-Cas9. Also, we can make or insert an enzyme to protect the virus, or we can make a protective coat to the virus, which protected from the immune system. A study has been done in, I think, 20 years ago for 3 patients of, I think, myeloma, and it was another cancer. So they tested on the solid tumors. At first, they figured out the CRISPR could stop the growth of the tumors, but after some time, it returned to grow back. So it was not a successful therapy for at that time. Also, further investigation is needed to approve this is 100% a real cure for cancer. Because also we need animal models, like mice models, for this kind, because we know that mice's genetics looks similar to human, but also we may need 10 years, 5 years, or longer, because also we know that even to a simple antibiotic to be approved, it needs a lot of time. This is something that may change the history of science and molecular genetics. But hopefully we hope that someday the CRISPR will be the right cure for the cancer-degenerative disease. We have another thing. What about the ethics? What about the ethical issues for it? Because someone may say that this is something, how can we change our genomes? How can we change it? It's not only in human, even in plants and animals we can do this. Because nowadays we can see so many fruits and vegetables that was not present 100 years ago. But how? Because they alter the DNA, the genes, the DNAs and the RNA, and they just edit it. Sometimes it's for a good thing, but also if we are not careful about it, it can be used as a biological weapon. That these person, the guys that want to do some fight, it's not only by real weapon, by biological weapon. They can just make a change in my gene or making something that's harmful for humanity and spread it. Like the risk of unforeseen and undesirable effects in clinical applications. Also, we may not have proper tools and items to do the gene editing properly. A matter of informed consent, the risk of exploitation of eugene. Maybe also healthy genes may be altered with this. What disease can CRISPR cure? We have a diagram here that luckily we could cure the leukemia, the solid tumor, the B-cell cancer, HIV, and so more with the help of this technique, which is very beneficial, like hemophilia, beta thalassemia, cystic fibrosis, Alzheimer's, and etc. The genetic diseases. What are the successful range? Is this a successful range or not? So, 42 patients out of 44 patients of the beta thalassemia were cured and were able to live a better life. And also, 31 sickle cell anemia patients were free of the symptoms, and all had been previously diagnosed with severe cases. Also, effective strategy for the treatment of cancer and many other diseases. We can also use this treatment for other diseases.